Hi everyone, and welcome to tissues. So tissues are where life, or formerly just single-celled organisms, start kind of joining together, forming a lattice, a network, and becoming what we now call a tissue. Humans, since we're studying human anatomy, have many different types of tissues. So we're going to start with a basic introduction of this concept and its origins. So a few definitions. Um, the first thing is the body is huge, quite frankly, in terms of biological, in terms of all organisms. Um, so in order to maintain such a ginormous, long-lived single organism, what's happened is groups of cells have had to multiply lots and lots and lots of times. And not only that, but use the same genome, the same genomic information to turn into multiple cell types, right? So we're going to look at that concept a little bit more throughout our talks, but for now, let's just get some basic definitions. The first is that of the tissue. So a tissue is, a group of cells that are similar in structure and really location, and they perform a common or related function. And then we have also the study of histology. So histology is the study of those tissues. Generally, it's considered a microbiological field, so we'll take a look at how it's performed, so those processes. And also we have these four basic types, one of which my head apparently is sitting on top of. So there's epithelial, connective, that's the one you can't see, muscle, and nervous tissue. Let's talk about this idea of cells living together as groups. So initially, and for the most part, the majority of life on this planet exists in a single cell state. Prokaryotes, even eukaryotes. But we're multicellular eukaryotes, meaning we decided to team up and have lots and lots of cells that are joined together. Okay, so here we have our organization of life. So the cell, remember, is the smallest unit that can carry out function. It's the basic unit of life. It's like the atom. It's like a single Lego block, right? And so if you build a bunch together, they become what we call a tissue. And we'll talk a little bit more about organs, organ systems as we go along. But essentially, this is the foundational element that supports the organism, right? Which in this case, again, is multicellular eukaryote humans. Here's how that works. So we have over here some example of cells from an epithelial type tissue, which we'll look at again a little bit more. But what I want you to pay attention to are these little, what we call a junction. A junction is a meeting point, an intersection. And so at these places where cells meet and touch, what you find are different types of adhesion type junctions. They form lattices, networks, tether cells together, help them either stay really close, or in some types of tissue, like we'll see in connective tissue, they don't exist, but they do provide these large fields of matrices. Matrix, it's basically like every cell in the neighborhood. Instead of like this is apartment style in connective tissues, it's like they all have really large front yards full of secreted proteins. We'll take a look at that in a little bit. All right, so that's the concept of how they join together, how they structure their neighborhoods. The neighborhood is going to be essentially the tissue. So the four main types of tissue we're going to look at. So nervous tissue, muscle tissue, epithelial tissue, and connective tissue. So nervous tissue, we're going to find that in your brains and your nerves. We'll talk about that probably like 3/4 of the way through the semester. Muscle tissue is one of my personal favorites. So if there's a hallmark of animalia, I would say it's definitely got to be muscle tissue, one of the main ones. Epithelial tissue is an interesting one. So the epithelial tissue is meant to interact with the outside world. So how we are formed from the very early stages after fertilization is we're basically a tube. We're a worm. We have lots of other tubes inside of us. And so all of the linings on the outside of the body and the inside of the body, all of those tube structures inside and outside are lined with this epithelial tissue. Some epithelial tissues are dry and rigid and they protect in that way, but others protect in a different way. They are moist or they secrete. And so we'll look at those as well. Connective tissue is the one we're going to start with. Connective tissue is a pretty broad one. It has quite a bit of diversity within this category. Connective tissue is basically this: any tissue that connects to another type of tissue, organ, or system. It's all the support team. So these are going to be varied. So everything from rigid stone-like bones to fluid parts and blood itself. All right. So as I said, cells are a little small, and so most of the work for tissues has to be done at the microscopic level. Right, and there's this typical set of processes that you'll see over and over again. So the first way is you have to prepare it for the microscope. That means taking a sample, maybe it's a tumor that's been removed, maybe it's a different type of tissue for some reason, taking it, slicing it up, and looking at it under the scope. But there's a couple of steps in between, one of which is you don't really want, number one, the tissue to start decomposing once it's been removed from the body. That doesn't help. So you need to fix it. Also, this fixation process can include linking proteins together so they stay in the same shape as they would have stayed more or less inside of the body when it was a living piece of tissue. The next section just means it's cut really, really thin slices. Sectioning's just how we decide the thickness for either what kind of stain we're going to do or what type of imaging. And there's a lot of options for that. We'll look at one of the most popular, at least one of the ones you'll see in your textbook over and over and over. The last one is staining. So this provides contrast because most of the time when you pull tissue out of the body and you put it on a slide, it's, you know, one cell layer thick, maybe a few more, but it's clear, quite frankly. You don't have a lot of natural pigments other than the ones that are used to deal with the essentially UV light, right? So we have to add artificial stains into it, and we can do it a number of different ways. We're going to look at the H&E stain, but ones that I've used I love are things like the fluorescent stains, which are antibody style stains. Also there's transmission, electron or scanning, microscopy, really exciting ways to look at the topography of tissue. So here is kind of a visual, I guess, step-by-step part of the process. Right here, this is a tissue sample. Just imagine we removed a tumor of some sort. We got to figure something out about it. Number one thing we want to know is it benign, or does it have a different destiny, possibly cancerous one? So the first thing we got to do is we got to fix it. So this fixation step can include a lot of different processes. Sometimes we literally just put it in wax. It's called paraffin wax, but it feels a little bit like candle wax. And it stays in there. Sometimes we simply just put it in alcohol or methanol. Some are fixed in formaldehyde, anything that's going to just stabilize those tissues and also prevent decomposition. Big important part of that process. The next one is slicing it into little strips, really, really thin. Sometimes they're only one cell thick, sometimes they're a few, but it's so we can put them onto slides and view them under the microscope. The next part is the staining process. The staining process that you're seeing here with the little Y's that have a pink bunny rabbit tail on them, that's reflective of an antibody stain. In this case, I would imagine maybe it's an immunofluorescently tagged antibody. So these antibodies are very specific. They're only going to bind to actin. They're only going to bind to myosin and nothing else. So you can get really specific imaging and in beautiful color, might I add, of the structures of single cells on the inside. Isn't that great? So the next part is you've got to haul all of those puppies over to the microscope. And there's a whole lot of different microscopes you can use. Some will use a confocal, others a light microscope. Most of what you'll see in histopathology labs are going to be just simply a light microscope. Others do automatic scanning and imaging. So whereas most of the time a pathologist will look at, and we'll see in a minute here, a little example, the tissue and just kind of eyeball it and look for morphology or the shape and arrangement of those cells. Other times we can put them through special cell sorting and counting type processes. Basically, a computer shoots a little laser beam over it and is able to count all of the nuclei of all the cells, or all of the, yeah, I don't know what you would otherwise count. I think that sounds pretty easy, but maybe, I'm sure cell bodies. Anyway, so we're able to put into a quantitative set of data set and take a look at, are these a lot of cells that are non-tumor? Are these cells from a tumor? Right, and take a little bit of a look at that. So over on this side, you're gonna hear this one and you're gonna feel this so much. It's the pink and purple blobs, right? It's the picture of tissue. And although we think of tissue oftentimes as being pink, it's really not. Remember, it's kind of clear. So we have to add stuff in. Okay, so this is eosine and hematoxylin stains. So an H and E stain is what they are. They're relatively inexpensive. You can run it pretty quick. Everybody does it. So an H and E stain works like this. Hematoxylin stains things that are acidic, right? So things like membranes, for example. And so it's going to, and DNA, right? So it's going to stain those structures blue. So you can see here's the nuclei of the cell being stained bright blue, so that's nice. But eosine stains basic structures, and it has a nice pink stain to it. Neutral structures oftentimes don't take up stain at all, right? Sometimes that's space between cells. Sometimes that might be something more neutral, like just water or accumulations of other neutral substances. And it gives us a basic idea of what it looks like. So are all your cells pink and purple? No, it'd be nice if they came pre-stained, but most of the time you have to do it yourself or just hire someone. So here's an example of how you can use those types of stains. Oops. Okay, so here's an example of how you can use those stains to start looking at tissue and seeing, is this healthy tissue or something more sinister going on here? So on this side, we have an example of what we call normal tissue. We'll see a lot of these later on. Okay, hyperplasia is when we're seeing things that are starting to get out of arrangement, right? So the cells are changing their shape altogether, they're arranging a little bit differently, they're clustering. So they're going from a normal state into a different state. You can see this is a transitory state. There's transition going on. Dysplasia means that things are not in the right place. They are displaced, right? So now we've lost this layer up here altogether. We know now that these cells are maybe, oh, and here's one right here that is mitosing, this one that looks like it has an egg that just cracked. Oh, I can almost get it. There it is. Yes. All right. It's tiny. It's a pretty good job. So these are cells that are undergoing rapid mitosis. As you can see, all these nuclei are really, really enlarged, and the cells are super big, and they're kind of sloppy looking, right? These are cells that are leading to possibly this next state, which is a cancerous state. Cancer is the unchecked cell growth in a tissue. It means that mitosis is not stopping, and that these cells are constantly and rapidly dividing. It means that these cells also aren't doing their jobs right because they're so busy concentrating on dividing that they are no longer doing things like secreting the right hormones or possibly protecting the cell from or the organism from invaders anymore, or digesting, or being able to do any of the number of things that the different tissues do in the body. They're too busy focusing on making new cells. They're not focused on doing their job, and they can clog up the system. That's one of the reasons why cancer is so dangerous. So look, you just spent some time learning histology, and you actually looked at the differences between normal all the way to cancer cells. You looked at an example of an H and E stain, and you know a little bit more about how cells work. Let's look at another aspect. Development. So we all actually do start off as single-celled organisms. What do you know? Ontogeny recapitulates phylogeny, classics, right? So after fertilization, the cell begins to divide. It divides regularly until it reaches this 8-cell compacted morula phase. So as you can see, going from a single-celled organism all the way to a multicellular organism. That is a funny looking arrow. There it is, okay? The next thing that happens is it starts arranging itself properly, because remember, we've got to make tubes. We are a tube. We're essentially a big worm with a bunch of tubes in it. Just think about how long your intestines are. If you were to stretch them out from end to end, it's really but the arms and legs have been nice. Anyway, so moving on, we start specializing right about here. We start moving cells into particular areas and regions. We start changing the way their shapes are. We're still using the same genome. Every cell in your body has your genome, only the same set of genes, but the difference is they turn some up high and others down low. And then a different tissue will say, oh, I'm going to use this group of genes right here. I'm going to turn those up, but I'm going to turn these other ones down low. And that's how you get all the different shapes that cells can have and have all the diversity of tissues. So moving on, after a little bit of time passes, we start specializing these tissues in the first three true layers. They're known as the germ layers. So the ectoderm, outside, mesoderm, middle part, and then the endoderm on the inside. So here's a picture of it. That's a little bit easier to look at. So what's neat is, although at this stage of development there's only a few different tissue types, literally there's 3, all of these different tissue types are going to be what we call the progenitor types for all of the big types of tissue and the huge amount of diversity that we have after we're born and then eventually mature into adulthood. So if you look here, the mesoderm, use red for it, muscle, connective tissue, all the meaty meat parts are in the mesoderm, or they come from the mesoderm. And then over here, the ectoderm, oops, I went right through it. Ectoderm becomes, believe it or not, your brains, right? So it's nervous tissue, but it's not just nervous tissue. We'll look at that in a second. And then finally over here, the inner part, right, or the endoderm becomes the inner part of your body. Things like your tube organs, right? Digestive tract, for example. So all this is going to fold in on each other in the completion of that tube shape as that embryo continues to develop. The other thing is, this is an interesting one, is how important it is to interact with the outside world, the epithelium, the outer side of the outside of the body. So it can be the skin or the epithelial layer of organs, right, the inside of your digestive tract. All 3 of those are going to develop into that epithelial layer, a very ancient, very important part of all of the tissues of your body. Well, not all of the organs of your body, not all the tissues, because they're all the epithelial tissue. All right, so again, the last thing I want to talk about is the fact that you're going to be looking for 4 main categories. 4 main categories, okay? The first is going to be the nervous tissue, right? So that's your brain, your nerves, your spinal cords, all the sensory type tissues. Oh, can't get my highlighter on. There it is. Okay, the next one is going to be muscle tissue, right? So the big meaty meat part of your body. A huge amount of your mass is exclusively muscle tissue. And again, one of my favorite tissue types. Anyway, we'll get to that later on. You know something's up when some biologist says that's my favorite tissue type, of course. Anyway, we'll save those exciting things for later. The next one, and I think one of the more difficult ones, are going to be the connective tissues. The connective tissues have a lot of diversity to them. And we'll spend some time. Remember, that can go everywhere from bones to fat to blood cells. Of course, things like your tendons and your ligaments. There's a lot to it, and we'll look at all of it. And then finally, what color haven't I used? Oh, hey, that one's good. Okay, epithelial tissue, right? So these are where all of the surface parts, the hollow insides of the organs, how things are secreted, the outside of your body and skin. So anything that has to kind of be on the outside surface of an organ or the organism in general. All right, the epithelial tissues. And we'll look at a couple of special cell shapes in an upcoming video. All right, well, I hope you enjoyed our talk about the introduction to tissues. And if you have any questions, please feel free to email me anytime, okay? And if I don't reply right away, just email me again right away. All right, we'll have a great week studying.